and externally, okay, and play a decisive role in the interactions of humans with the environment. So just to give you a few numbers, we harbor as many bacterial cells as human cells, and this represents a genetic repertoire that is 100 times larger than the genes in the human genome. And among these different, sorry, and among these different niches, the gastrointestinal tract is the most densely populated ecosystem and the most extensively studied. How we study this ecosystem? So basically by sequencing the DNA content of fecal samples derived from a given individual, okay, by an approach that we call metagenomics, and that allows us to obtain a cartography of the microbial species present in this fecal sample together with its relative abundance, okay? So in terms of complexity of this ecosystem, by this analytical approach, we are able to detect a kind of in the range between 100 or to 500 microbial species, even if this is extremely variable across individuals, conditions, and populations, okay? And importantly, also to take into account is the fact that in the vast majority of these studies, what we have access is to a single sample per individual, is what we call cross-sectional studies that seeks to explore the compositional variation of the gut microbiome in a given population, on a given clinical group, or even in a given country. There are less numbers of studies, even that we have more and more in the literature that follows the dynamics of the gut microbiome composition of an individual over time. Okay. And the main questions that we want to address in this kind of analysis are first, who is there, that we answer with these taxonomic profiles, what they are doing, that we answer with the projections of cellular function associated to these microbial species, and most importantly, what does this mean, this composition of the gut microbiome in different study contexts that we address by mean or the statistical analysis and machine learning algorithm, or it's the framework most interesting here by different modeling approaches, okay? In terms of statistical analysis, it's common to observe in this kind of studies a battery of hundreds of potential biomarkers that we find significantly associated, biomarkers derived from the gut microbiome profiles that are significantly associated to one or several clinical groups by means of different statistical approaches. So this is one example of one of these studies that we were involved in, derived from the Metacardis project, where the goal was to identify these biomarkers in different clinical groups of individuals that corresponds to different levels of severity of this ischemic heart disease, okay? So we end up with this battery of microbial species here in blue, and also serum biomarkers in the blood here in red that are significantly increased or decreased in one or several of these clinical groups. And also by using different machine learning methods, we have observed in this study, but also in many other studies, that these biomarkers derived from the gut microbiome harbors a higher predictive power to classify samples in different clinical groups, like is in this case, that models trained with traditional clinical variables or models trained with other types of omics data, like for example, variations or polymorphisms in the human genome. But the problem is that the biology behind these associations are extremely hard to distill simply by this kind of statistical analysis. The mechanistic insights are hard to distill, okay? And it's important to take into account that what we get in this readout of the gut microbiome is just a snapshot of a much more complex ecosystem where the microbial cells can move and interact across between them across the gastrointestinal tract, and can also interact with the human host, providing key metabolites that we humans are not able to synthesize by ourselves, like, for example, short chain fatty acids that will emerge in several of the following slides. And many of these interactions are driven by different metabolic capabilities of these microbial species that could drive or competition relationships for nutritional resource, or even mutualistic interaction, like in the example here below, where microbial species exchange metabolites in order to sustain cellular growth. Okay. So is here where the modeling approaches are interesting in order to understand this microbial ecosystem. And the approaches that has been applied to study the human gut microbiome varies in terms of the level of parameterization of microbial growth rates and parameters of the human gastrointestinal physiology, and the consideration or not of the metabolic variability and the potential interaction that could emerge between community members. At one side, we have these metabolic agnostic approaches that does not take into account cellular metabolism, like, for example, the use of these generalized Lotka-Volterra models in order to study species-species interaction when we compute the relative abundance of microbial species over time as a function of the cellular growth rate, and also the pathways interactions with other community members that can be positive, neutral, or negative. So here, these interactions don't describe what are the mechanisms behind them, and does not take into account the metabolic flexibility of community members. At the other side, we have what we call metabolic explicit approaches, where we can find at one side kinetic models of microbial communities that some authors call as black box modeling of cellular metabolism, because they simplify this microbial ecosystem to a few representative and few metabolic activities of relevant for the interaction between the good microbiome and the host. And finally, we have other approaches that does an explicit modeling of cellular metabolism of all community members by the use of genome scale metabolic models. Okay. So just a single example of one of these kinetic models here in this paper. The authors start by collecting a group of variables that describes the physiology of the gastrointestinal tract in terms of the average length of different gastrointestinal segments, the flow rate of water entering the gastrointestinal tract, being absorbed by the gut epithelium and reaching the fecal compartment, and also the rates of contraction of intestine walls, and also the absorption rates of the gut epithelium for different compounds, okay? And also in parallel, they characterize the growth dynamics of just two microbial species that describes or that defines or that belongs to the main taxonomic group that compose the good microbiome, the Bacteroidetes and the Firmicutes, where they measure, for example, the rates of secretion of these different molecules, these short-chain fatty acids that I previously mentioned, as well as the growth rates of different microbial species. And with all this, they build a system of differential equations that describe the bacterial growth dynamics of this two-member community across the gastrointestinal tract, from which they are able, for example, to simulate the relative abundance of these two species under different uptake rates of glucose, in parallel with the rates of absorption of this short-chain fatty acid produced by these two microbial species by the intestinal epithelium. And also they are able to simulate the spatial profiles of these two microbial species along the gastrointestinal tract under a standard Western diet, okay? These models are extremely powerful, but requires an extensive knowledge of different physiological parameters in terms of cellular growth rates of different community members, or even the logic of nutrient use of these microbial species. And these parameters are extremely difficult to obtain for a large fraction of microbial species in the good microbiome, because many of these species cannot be cultured in vitro. And for those that can be cultured, most of these parameters are derived from monocultures. And this condition, the monoculture, does not reflect the real environment that the bacteria finds in the gut microbiome, okay? Also, this equation in these kinetic models, as I previously mentioned, tends to simplify the main functional capabilities of community members and covers only a part of this metabolic potential of the community, and don't allow to explore potential emerging metabolic properties of these communities in terms of species-species interactions that are derived from potential metabolic interdependencies. Okay. So it's here where this second approach comes up. In contrast with the kinetic models, this approach of using genome-scale metabolic models is based on the fact that the entire knowledge of the behavior of a cellular system can be derived directly from the functional annotation of its genome, by means of collecting what we call gene-protein reaction associations, which are basically the associations between genes coding for enzymes and the biochemical reactions catalyzed by these enzymes, okay? And these associations, this GPR association, can be directly extracted, as I mentioned, from the functional annotation of genome sequence. And these functional annotations, we have millions of them in public data banks, including dedicated repositories that store the genomic knowledge around microbial species of the human gut, okay? So with the ensemble of GPR associations derived from a single genome, we have what we call the genome scale metabolic network of the organism, from which we can represent this network in the form of what we call an stoichiometric matrix, okay? Where rows are metabolites, columns are reactions, and indexes are simply the stoichiometric coefficient of each metabolite in each reactions of the model. Positive if the metabolite is produced, and negative if it's consumed by a given reaction. With this stoichiometric matrix as a core structural element of the system, the main modeling approach that is applied is what we call constraint-based modeling of cellular metabolism, that consists in explore the space of feasible flux distribution across reactions of the metabolic model that satisfies a given set of constraints that we apply to the behavior of the system, the most important of which is to assume that the system operates on a steady state condition, which means that there is no net production nor consumption of any internal metabolite in the model, okay? And there are different ways to explore this space of feasible flux distribution. The most common one is to use linear optimization algorithms in order to find a single point, a single flux distribution in the solution space that maximize a given objective function, okay? And this objective function is commonly defined in terms of a reaction in the model that represents the biomass production of the system, and that represents or contains all the different elements like amino acids, cofactors, or vitamins, nucleotides, or lipid membranes that any cellular system needs in order to sustain growth, okay? So the most commonly used approach, for example, is flux balance analysis, which finds that this single flux distribution that maximizes biomass production. But there are other approaches like the parsimonious FBA that finds an alternative optimal solution that maximizes biomass production while minimizing the total sum of fluxes across model reactions, okay? So at bioinformatic level, there are many tools that allow us to obtain a draft reconstruction of these genome scale models directly from genome sequence. And we have even a dedicated repository that is called Agora that contains curated genome scale metabolic models in the latest release for more than 7,000 microbial strains of the human gut microbiome. And just to give you an idea of the size of these models, at an average size of 1,700 reactions or equations in the modeling terms, and 1,500 metabolites, okay? So with this element, with these genome scale metabolic models, the community models is simply to put together the individual genome scale models of all microbial species that we identify in a given sample for a given individual, models that will grow in the same environment that reflects the luminal content of the intestine, that receives nutrients from the diet, and that secretes compounds or chemical compounds to the fecal environment, okay? And here what is optimized by linear optimization is an equation that we call the community biomass, that is just the individual biomass of all community members weighted by its relative abundance here. So this allows us to evaluate the behavior of these microbial ecosystems from different individuals, here, for example, into groups of patients, healthy and individuals with Crohn's disease, under different environmental conditions, and have a readout of the potential for production or consumption of different compounds by the good microbiome. Just to give you a short example of how this approach could contribute to a better understanding of the biomarkers derived from a statistical analysis, I would like to introduce this chemical compound, which is trimethylamine, okay? Which is a molecule that is exclusively produced by the gut microbiome from dietary choline, okay, that comes from nutrients like, meat or eggs, and that is a metabolite that after in the liver is transformed to this second compound, trimethylamine oxide, that has emerged as an important biomarker strongly associated to the severity of cardiovascular disease. Okay, here we observe the dynamics of this metabolite in the Metacardis study, and we see this gradual increase in the blood levels of this compound across groups corresponding to different levels of severity of the coronary disease. Okay, so here what we get is the results of these simulations on the community models of 3 different patients or individuals under 11 dietary profiles. Okay, so we have this readout of all the metabolites that can be produced by these different community models under these different nutritional conditions. And if we focus on this particular metabolite, we can observe that the production profiles strongly varies across these 3 individuals. And we have this patient 3 for which the potential for production of this trimethylamine compound is the highest, no matter which is the dietary contents that we get the simulation. So it's something in the microbiome of this individual that is behind this phenotype of trimethylamine overproduction. And we can also extract from these results the complete network of cross-feeding interactions that is behind this phenotype of trimethylamine overproduction. Like in this case, for example, this is consequence of the presence in the microbiome of this individual of 4 microbial species that takes choline from the diet and produce this trimethylamine compound in combination with other 4 species that are able to take this compound and transform to trimethylamine oxide in the context of anaerobic respiration. Okay, so allow a mechanistic understanding of why we have this phenotype in the microbiome of this particular individual. So this modeling approach has also many drawbacks. Okay, first there is a strong dependence of the completion of genome sequence to have an accurate genome scale metabolic models. Also, this biomass objective function that is at the core of the modeling approach is very hard to define experimentally. And the strategy that is followed for many of these genome scale models is to adapt the biomass equation of model organisms, like, for example, Escherichia coli, with minor variation depending on the cellular characteristics of main bacterial lineages. Okay. Also, this maximization of cellular growth in terms of biomass production, that again is at the core of this modeling approach, is not always true. And the fact that it's focused on metabolism makes difficult to take into account other environmental factors like pH or osmotic pressure. And most importantly, in the context of microbial communities, this community biomass equation is a challenging concept in the sense that in the approach that I show you is somehow that is fixed, that is derived from the taxonomic profile of the microbiome of each individual. And for example, with this framework, we cannot simulate directly how the composition of a given set of species will evolve under a given environmental condition. Okay, and also does not take into account the spatial distribution of microbes in the ecosystem. So it's here where the individual-based modeling could contribute to improve these different analytical aspects. And in fact, there are few approaches, individual-based model approaches, developed to study these microbial communities of the human gut. The few that are available combines a core of modeling that is this COBRA approach, constraint-based modeling approach, on genome scale metabolic models by iterating this simulation, this flux balance analysis simulation, updating at each time step, let's say, the media conditions, and also simulated the spatial movement of these microbial agents in terms of these genome scale models on a two-dimensional grid space. This is an example of one of these approaches that is called Baccarina, where here agents are microbial cells, and each agent is represented by a single genome scale metabolic model that acts in this 2D grid space based on a list of rules that has as at core of the modeling approach this constraint-based modeling framework that defines the rates of agent growth or agent death based on a threshold, defined threshold in terms of biomass production, for example, and that is parameterized from variables extracted or derived from the literature or from reference database. So this is an example of the modeling framework that you can obtain with this approach. Here, the authors model a microbial community of 7 species with 200 individuals per species that puts to grow into this, what they call arena, under 2 different types of environmental conditions. A uniformly distributed rich medium and a rich medium, but where a specific compound, which are the mucus glycan, shows or follows a gradient of abundance from the bottom to the top of the grid that reflects the luminal profile of this metabolite in the human gut that is close to the gut epithelium and decrease in abundance toward the lumen of the environment. And it's observed that the behavior of the community differs as is expected. And in this framework where we have this mucoglycan gradient, we have that the proliferation of bacteria, like Bacteroidetes theta-Omicron, that can grow on this particular carbon source, but is concentrated in this region of the grid space and show the best behavior in terms of cellular growth in comparison with other community members. And you can also, from the results of these simulations, extract these production profiles of community members for different chemical compounds, here again short-chain fatty acids, from which you can infer this network of cross-feeding interactions between community members based on the consumption and production of these metabolites. There is this second approach also that is called COMETS, and that is similar in concept to Baccarina. The difference is that here the agents are not microbial cells, but the whole microbial populations, and that simulations can be done also on this 2D grid space, but also in conditions called well mix, that is similar to the previous modeling framework that I explained in previous slides. So this is an example of the results that you can obtain with both approaches in a simplified community of 2 microbial species, where you see that this grid in the 2 different methods follow the same topology in terms of abundance of, at one side, bacteria that produce hydrogen, this Clostridium beringi in red, that grows in the region with overproduction of hydrogen, and the second bacteria, Archaea metagenoma, this second archaeal species that grows in the region space where there is this overproduction of methane. Okay, so it's here that we would like to introduce or to see how this modeling framework could be implemented in Gama. So the idea that we had in mind that we would like to explore is first to implement some data integration modules that will allow to import all this catalog of genome scale models and quantitative microbial profiles that are used to define the community model at one side and the nutritional profiles that defines the environment. And in a second step, being able to implement this constraint-based modeling framework in Gama in order to address these different ways of modeling the microbial communities with a particular focus in this individual-based community modeling. With some questions that emerge that we would like to discuss or potentially to have some feedback in the sense of the concept of agent in this modeling framework. It is better to consider microbial species or microbial cells or even the whole microbial ecosystem of a single agent. We don't know exactly what's the limit. And also the level of complexity of these individual agents of models that can be handled in Gamma. For example, for bacteria, we have that these models has 1,500 reactions, but we can integrate in this framework the metabolic model of the human that is much more complex, around 30,000 reactions. Also, the fact that nutrients in this framework can be considered of agents or as agents or not. And finally, if we can go beyond this tooth grid modeling by considering, for example, the 3D structure mimicking the topology of the good ecosystem. So with this, I finish, and thanks all of you for your attention, and don't hesitate to ask.
Well, thank you, Edi, for this presentation, this keynote. It was very different from what we are used to, so it's
That's
keynotes, and thank you for
that.
that. So do we have questions already? So as I told you, maybe we have to be a bit strict on time, so not that many questions. Okay, so no questions yet. I have maybe some remarks or things for you. So it's not my domain, so I cannot talk too much, but from what I understand, okay, you're questioning about is it possible to switch from that model, which is math-based, to something more agent-based with Gama, for example. And I think you will face some problems of performance. It would be probably the bigger problem you will have is the number of agents and how you handle them. And there are a few ways to address those problems. And actually in the chat, right now we have 2 PhD students that work on 2 different ways to manage performance of models with Gama, with different approaches. One which is more about the modeling approach, and one which is more about the computing power. So I don't know if they want to say something about it, but maybe it could be interesting. And I see we have a question in the chat. Do you have experiment in building models with stakeholder, biologists, and Gama could be a tool to open discussion?
I will say that this is the framework because I'm biologist by basis, so that's why I'm interested. I build these models from these metagenomic profiles, but as I mentioned in the presentation, they have many limitations, particularly in this dynamic modeling of communities, and yeah, that's why. I thank you for allowing me to post the analytical framework and see your insights about ways to give little steps toward integration into GAM.
Yeah, time is running, but I will take the last one. So we've got Arthur, is one of the students I was talking about, who says, since you're planning to integrate different tools at different levels, cellular, genomic, do you plan to use Gama to work and explore interaction between those levels?
Yes, absolutely. Yeah. I mean, the simplest, we would like to start small. So if we arrive to do these simulations on few genome scale models and see how it behaves in terms of few agents of this type, microbial species. That will be great, but this opens, as you say, a scope of applications, including integrating the human genome in the modeling framework and exchange between bacteria and humans, and different layers of complexity of the cellular system in terms of transcriptional profiles, proteomic profiles, etc. So yes.
Okay. Well, thank you again for your presentation. And unfortunately, I have to start with the short presentation now. So next one will be Saeed Soltani, I guess, presenting probabilistic tsunami evacuation modeling for City of Catania, Eastern Sicily. Saeed, we can see your screen. We can hear you. So whenever you're ready, you can start.
Okay. Hello, everyone. I'm Saeed Soltani, and today I'm going to talk about the probabilistic tsunami evacuation modeling for City of Catania in Eastern Sicily. And first of all, I should say that I'm not a computer scientist or software developer. I'm a seismologist. And recently, I am a visiting researcher at the League Laboratory in University Grenoble-Alpes. And thanks to Julie Dugdale, I have a little information about the ABM and Gama platform. So actually, I'm here to learn from you rather than showing some fancy results. So, yeah. I'll start with who I am. I'm working in the European Plate Observing System, which is a multidisciplinary distributed research infrastructure that wants to integrate the data and data products and other facilities of earth science community in Europe. And if you are interested in the data set, you can see this, you can use this QR code to visit our website. And actually, the EPOS allows researchers to access the fair data and dataset from various earth science disciplines in Europe. The EPOS is divided into 10 categories, and we are working in the tsunami thematic core services, which aims to establish the sustainable and harmonized service for tsunami science, and specifically for tsunami risk reduction. And we do that with the accessing to the different data products, software, like workflows and other services in the European level. So the TCS Tsunami are organized within the 4 pillars. And actually, I'm the manager of the European Tsunami Risk Service, which is part of the pillar 4. And if you are interested, you can visit our website for more information. So since we have a lot of we have large amount of the hazard curves, hazard values, for example, and risk products like the vulnerability curves, like the economic cost and loss curves, but still we have lack of any information about the evacuation in Europe because we don't have any real data for evacuation in European parts. So we decided to have a framework, this tsunami coastal framework, or what we call the TSEM. And for the case study, we choose the city of Catania, which is a big metropolitan area in Italy, which has prone for the big tsunamis, and it's very close to the large volcanoes. And also it has the last experience of the tsunami in Europe. And we have right now a detailed and accurate tsunami scenarios in this area. So with this data, we want to, for sure, we want to have a population response to agent-based modeling. And three objectives important for us is to have the presence or absence of the tsunami warning system in this area and characteristic of the people, which we focused on the elderly people with disabilities, which lives in the beach houses in Catania, and also to see the level of awareness and the perception of the population about the tsunami risk, to finally evaluate the tsunami evacuation risk, which means the probability of safe evacuation in this area. when we talk about the tsunami risk, we think about the probabilistic tsunami evacuation modeling, which can be defined based on this simple equation, where E stands for the safe evacuation, and theta represent the vectors of uncertain parameters. For example, the tsunami source parameters that we know about the uncertainties, and the parameters related to human response and decision that we don't know. And we want to calculate the probability of evacuation given by theta that can be approximated by the percentage of people that can safely evacuate, which can be modeled through the ABM. And finally, the probability of safe evacuation can be estimated easily by some Monte Carlo simulation scheme. It can be estimated by average of over all the scenarios. The idea of the system is very simple. So we have the earthquake that can happen all around the Europe. Then after the earthquake, we have some arrival time of the tsunami. That arrival time can be 2 minutes if the earthquake can happen in the Calabrian arc near the Catania, or it can be extended to 1 hour depending on the earthquake source. For example, in the Cyprus arc, if it's happened in Cyprus arc in southern Turkey, the times it's like 1 hour. So in this time of the arrival time, because people have no perception about the risk, there is no reaction. For example, if it's the night scenario, it's no move or regular move in the day scenario. And then we have an alarm, which is the alarm generally starts at 8 minutes after the earthquake based on the data that we have from the civil protection in Italy. And after this alarm, we have a reaction from the people, and the reaction time is extracted through the questionnaires. And after a while, we have the wave of the tsunami that reach the beach, and the evacuation continues. So it's simple as it. The advantage of the RT SEM model is the hazard values. So for example, for the old simulation of the tsunami, we always have one source parameter that, and then we extract the flow depth and the inundation map in the beach. But now, based on the new simulation all across the Europe, you can see that from Turkey to the Caribbean arc, for example, based on these all of these source parameters, right now we have 4,200 initial scenarios that we reduced it to 300 unique scenarios for that can affect the city of Catania. And right now I'm focused on only 23 key scenarios. The limitation of this large amount of hazard values is that, so if we have, for example, 300 hazard scenarios, we need 300 runs for each ABM scenarios. And when we, for example, when we change one parameter in the scenario, it goes to 600 or 900 hazard scenarios. And also the number of layers for each 4-hour simulation of hazard scenarios, it's 480 layers, because we extract the data in every 30 seconds. So for 4 hours, it's 400, which is a lot of. So if we want to model all of these scenarios, it's like it needs a lot of resources, and also we should be mindful about the carbon footprint. So this is the limitation of the model for the moment. Anyway, the other advantage that we know the alarms very well in this area. So based on the different experience from the previous earthquake, we know that the alarm of the tsunami is like about 8 minutes, as I said before. So this is the example of the earthquake that happened in Turkey, 7.9, and issued a tsunami alarm in Italy. So we can use this constant value in our model easily. And about the agent parameters, as I said, we focused on the elderly people in the Sicily, which has one of the most populated area for elderly people in all Europe. And we did 2 different questionnaires in this somehow in this area. The first one, the tsunami risk perception in central and southern Italy, about the perception of the risk, that we received 156 questionnaire. And the other one done by UNESCO, it's the tsunami evacuation perception, which is done in the southern Spain with almost 500 questioners. So we extract the data from these questionnaires and to extract the reaction time. So for the reaction time, we check the emotional and behavior reaction of the Italian people, the real reaction of the people from Umbria March earthquake, and also the survey from Chipiona in Spain to consider these values as a human behavior in Italy. And then we consider normal distribution for each human behavior, considering the range of evacuation time between 5 to 30 minutes based on the real evacuation time for people in Southeast Asia with the real data that we have, for example, in Indonesia. And then we fit the different possible distribution for this reaction time. We choose the log normal, which is the common one for the hazard that has that involves some delay, like the tsunamis or the volcano eruption. This is the very simple diagram of our model. We have environment building that is affected by the tsunamis, content of the people with the different gender and disabilities, and the different actions. And we define the safe areas at the vertical shelters within the buildings, with the highest floors, and the horizontal shelters 5 kilometers away or 40 meters above the sea level. And all of this environment has type of early warning system. So for the base model, for the 23 model that I told before, we did our modeling. For the base model, we have no early warning for the moment. The wave threshold for drowning is 1 meter, which is more or less the height that people drown. The number of agents in 1,170, which is the 1% of the real city population. And this is the characteristic of the different scenarios. You can see that from the magnitude 7 to magnitude 8.2 with different slip rate of the source parameter. So we did this simple simulation here, and you can see that starting the simulation, there is no hazard here. Some people arrive early because they are, for example, located in the buildings very close to the shelter. The shelter is the green area. And then after some cycle, we have the tsunami waves here. And you can see that some, you can see some people drowned and you can see the casualties over time for each scenario. So we redo it 23 times with different scenarios, but with the same scenario, different hazard scenarios, but the same agent scenarios. And here it's the results. So you can see the casualties per time. For example, for the very small one with the 7-point magnitude, you can see that only 40 people are died, but it goes up to 300 people. The safe evacuation percentage is about 97% to 75%, and the probability of safe evacuation based on the simple formula that I showed you before, it's about 82%, which is not very bad. And that's it, and the standard deviation is 0.045, which is a reasonable value. And about the future works in Gama code, right now I'm working to assign family attachments and car evacuation strategies with help of Julie. And the data set is already down, so we are going to do it very soon. And the most important part for me is to optimize hazard inputs by applying, I don't know, some optimization algorithm, or if we had to, by reducing the data layers. And then think about to set the number of scenarios, because as I said, if we change only one parameter, we should do another 300 scenarios, and running this extensive simulation, we should be mindful about the carbon footprint in this part. And about the risk assessment, okay, we have some future work, but it's not the scope of this presentation. And finally, any comments, suggestion, and more important, the interest in contribution are welcome. So if you are interested, you can just send an email to me for more contribution. Thank you.
Yes, thank you for your presentation. I see we already have one question in the chat for you. So Luca is asking, is 1% of the population enough to represent the behavior of the population in the area?
Actually, I'm thinking to do it as more as possible realistic, but as I said, it's like if I want to do that in 300 different simulation with the 100,000 people, which is the real population, it's very expensive. And I think I should find some reduction in the hazard model and then think about how to distribute the agents. So the main question right now is if we somehow reduce the hazard part, the needed to resource for the simulation of the hazard part, so we can do it in another way for the population.
Okay, thank you. If I can add something is, okay, time is running, but I'm just saying it. So you're talking about, in the end, using cars, integrating cars, basically. And have you thoughts maybe about the multimodality, so people using different means of transportation? Maybe that can be something interesting.
Yeah. What we think about it is just one thing that we think about to like some to have at the end some hourly different evacuation, which means that, for example, it's in the if it's happened in the 5 p.m. when people I don't know, or maybe in the noon that people are in the beach, so it's different. So and then we can consider the car evacuation that it's again depend on the traffic, so the different hourly evacuation can be affected by the car evacuation. So it's the thing that it's that I'm thinking. So and I'm in the beginning of that. I don't know. Yeah.
Okay, well,
Thank
once
you.
again, thank you. So Patrick has one more question, but maybe you can answer in the chat directly because we need to switch to the next speaker. So next come, I don't know who's speaking. Maybe is it Eva for the playground Wars Territorial? Yes. Then if you're ready, we can hear you already. When you are, you just share your screen and.
Okay, so good afternoon, ladies and gentlemen. My name is Ewa Tokarska-Gołębiewska, and with my co-author and supervisor, Dr. Maciej Szarejko, I would like to present to you our territorial conflict simulation model. In our project, we developed a simulation model to examine territorial conflicts within social groups, exploring how those dynamics can influence the design of conflict-resilient urban spaces. Just a quick peek into my presentation plan, and as we are short of time, I will just skip it to the research objective. So territorial conflict and social interactions are fundamental dynamics in urban spaces, shaping the way communities function and develop. As we know, urban spaces aren't just physical environments, they are social environments when people interact, form connections, and sometimes came into conflict. Territorial behaviors, whether subtle or overt, are a part of human nature and can influence everything from how safe people feel to the overall harmony of a neighborhood. The primary objective of my research is to create a simulation model that provides insight into those social dynamics, specifically how interactions between individuals in shared spaces can either lead to cooperation or to escalate into conflict. By analyzing these patterns, we aim to uncover design strategies that could actively foster collaboration, reduce conflict, and ultimately make urban areas more resilient to social tensions. Our starting point was to examine the dynamics of playground wars, the kind of territorial conflicts that arise even in small similar spaces where children play on a day-to-day basis. This concept allowed us to examine smaller scale environments that resemble real-world neighborhood country yards and playgrounds found worldwide. In these aspects, we observed how many networks of friendships, social connections, and family ties influence interactions and conflicts. An important aspect we consider was the influence of family models, particularly in Western European context, which often contrast with the structure of migrant families. These families frequently cluster with close residential proximity, creating tight-knit, interdependent communities. This proximity and collective presence can strongly shape interactions, both fostering resilience within the group and at times creating tensions with surrounding communities. Understanding those dynamics offer valuable insights for designing urban spaces and accommodate diverse family models and foster cooperative interactions. Regarding the theoretical foundations, the model utilizes agent-based simulations techniques to explore how interpersonal interactions rooted in individual experiences and spatial awareness, biological concepts such as kin selections, I mean by that the Hamilton rule, as well as trusted network, and by this I mean the number numbers, influence collaboration and conflict within communities. Regarding the input parameters, so the model's input data include predefined agent parameters such as gender, so G, initial level of collaboration, LC, level of danger, LD, base of kinship, base of Dunbar numbers, as well as base of known spatial elements, which is BSA. And those parameters evolve dynamically over the course of the simulations. As agents move through a shared environment, then they continuously expand their knowledge of spatial elements. So this is the base of known as spatial elements, BSA, and update their LC, level of collaborations, and LD, level of danger. And it is based on their individual experiences, which is creating an adaptive feedback loop for us to analyze. So regarding the model itself and its structure, excuse me for a second. So a key feature of the model is the agent interaction process triggered when two agents encounter each other within a specified proximity. This interaction, referred to as situation meeting, initiates a sequence of cognitive and behavioral processes where both agents, X and Y, assess the interaction based on their prior experiences and existing perceptions. Each agent perceives the outcome of the meeting differently, producing an individual meeting outcome, IOMO, that can be either positive or negative. The subjective nature of those interactions allows for divergent outcomes for the participants, even from the same encounter, and this is very important in terms of perception. The individual meeting outcome plays a crucial role in shaping future interactions. The agents adjust their attitudes towards subsequent meetings, influencing their level of collaboration and level of danger parameters. This process not only affects their interactions with new agents, but also alters their perception of agents allegedly cataloged in the base of Dunbar numbers, so already known other agents. As a result, the model simulates the evolution of social relationships and the potential escalations or the escalation of conflict over time. And I believe regarding the overall view, it would be better to show it to you through the scheme. So as already mentioned, the input parameters for the agents are set at the beginning of the situation, and in case of the proximity situation meeting encounter, then we start our meeting. And each of the agents defines whether they know or not the agents they are currently encountering. So if the Dunbar number is within 1 to 4, then it means they already know them. If it's not, then they go to the 5, which means they not know it yet. It is crucial that the assigned path and the region where the situation meeting occurs can influence how the agent will perceive the meeting itself. So if later on we check whether the agent we are meeting is within our kinship area. So A, which is the number of the meeting, is bigger than 0. If not, that means that the agent we met is only in our kinship model, it is only in our kinship base. And if yes, then we evaluate whether the number of positive or negative meetings were good from our perspective or bad. If that, then we add a certain amount to our collab level. And we can also decide whether we have certain preference towards the gender. For example, 2 girls can with bigger possibility like each other just because they are 2 girls. And the same for the opposite gender. And then in case of the negative meetings, the elements are the same. Regarding the situation where we do not know the person, we again come back to the gender issue. And in case, and this is happening at the same moment for both of the agents, which allows us then to compare the agents' X and Y levels. This would define whether the meeting is positive or negative according to the factor M. And at the end, we actualize all of the aspects regarding the number of meeting, whether it's positive or negative, the path level, collab level, as well as danger level. And that ends with the end of the meeting, which brings us to the beginning of the loop. And now if I can. So regarding the conclusions and practical applications. So in practical terms, this model might provide planners with a tool for designing socially resilient spaces. Rather than simply focusing on aesthetic or functionality, we may now have a way to anticipate how people may interact, form bonds, or experience conflicts within a given space. This allows us to create an environment that not only accommodate diverse communities, but also foster social cohesion and minimize potential conflicts. The presented model provides a valuable tool for examining the dynamics of spatial conflicts and its spatial context by analyzing different interactions and development of territorial behaviors. The model can be applied to design conflict-resilient urban spaces as well as communities. The insight gained from the simulation can offer urban planners and designers a deeper understanding of how social conflicts emerge, how they spread, and how spaces can be structured to mitigate tensions, fostering healthier, more collaborative environments. Research itself bridges the fields of architecture, urban planning, behavioral psychology, and biology by integrating knowledge from human sciences into spatial design methodologies. It highlights the importance of considering human behaviors in the context of territoriality and space, contributing to the development of environments that provide social cohesion and minimize the potential for conflict. Through this model, we aim to provide architects and urban designers with an innovative tool for analyzing and improving the social dynamic of urban spaces, ultimately contributing to safer and more harmonious communities. And additionally, I would like to add that my team, Smart City Hub on Wrocław University of Technology, is eager to start scientific cooperation with other researchers. We are currently preparing proposal for the NATO Science for Peace and Security Program grant. And please feel free to reach out and contact us regarding this or any other scientific cooperation, as we are quite a strong team during today's present conference. And in conclusion, I would like to express my gratitude for your attention. And now I invite any questions you may have.
Thank you for your presentation. Again, different from the others, so it's a bit refreshing. So do we have questions? Yes, we have questions. So Benoit is asking, do you have even preliminary simulation results?
At the moment, we are only expecting it, as we just created the model as a graph, and we are now in the process of putting it into the Gama platform and trying to experiment with real-like simulations. Because as you could see, the parameters we included, they were at the moment rather set, just to allow us to create a wide range of final result that we would like then to set up as close as to real world as possible.
Okay, thank you. Personally, I have one very quick question. I'm not exactly sure what the Dunbar number is. Is like the level of familiarity with someone?
Yes, precisely.
And, okay, perfect. And for the Hamilton rule, I think I know, but I'm not sure. It's like the fact to cooperate with someone from your family or something like this?
Yes, that means that when you are in proximity, there's this base of kinship, and there is bigger possibility of altruistic or generally cooperating behavior if you are somehow connected. When you see, but
Yes.
It's tricky because also when you see any kind of familiarity between two agents, then you can expect that they are similar, so you want to be more cooperative towards them.
Okay, I understand. And maybe so I would just ask one last to open a bit the question. So from what I understand, your model is not really set yet. And have you considered maybe the group behaviors? Because we know that we can have like internal competition and then cooperation for competition with another group. It happens often. So is it something interesting in your model or not?
What we considered very valuable in terms of the technique, so agent-based modeling, is that you can actually go not from the general group, but from the individual perspective. So what we hope to achieve is that through individual scenarios, we can actually achieve it from this very bottom-up approach, not from general group to the individuals. We are very interested in the individual approach in this term.
Interesting. I also think that from the individual approach, you will find the group, but yeah. Okay, well, thank you for your participation.
Thank you very much.
Yes. And now we have the next one. So let me check the list. Is it, who is presenting? Is it Maciej Szarejko, maybe? Or
Great pronunciation, thank you. It's Maciej Szarejko, but main presenter will be Szymon Dziedzic. Stage is yours.
Okay, thank you. Hello everyone, my name is Szymon and I'm a PhD candidate at the WST here in Wrocław. Today I would like to talk about stress and more specifically proxemic stress. What exactly that means, I will explain later in the presentation. However, I would like to start with what led us to consider this topic in the first place. The modern world is facing many challenges, some of which I have listed. An aging society, individuals who are lonely and physically inactive are particularly susceptible to mental disorders. Interestingly, physical activity could help mitigate this. Did you know that moderate daily physical activity can significantly reduce the risk of developing lifestyle diseases such as diabetes or even depression? This raises a question. Why do people not engage in physical activity? When designing recreational spaces, we often focus on eliminating physical barriers, but we almost entirely overlook psychological and behavioral barriers. When dealing with ergonomics, it's hard not to feel that we often treat humans like those popular wooden mannequins, the ones with flexible arms and legs that can be bent to test different postures. But we should go further. Our wooden mannequins should have beliefs, desires, and intentions. Our wooden mannequins should become real boys. When formulating problems based on observing people in built environments, we can use a component that analyzes advancements in health and anthropology to create convincing human phantoms that utilize our solutions even before they come to light. We can then iterate through successive solutions, ultimately leading to the development of the best possible design. Our model works in a fairly simple way. We introduce users into the analyzed park, allowing them to move freely within it. The park contains obstacles that narrow the field of vision. Additionally, the analyzed agent has human distance set, which I will explain shortly. As our walkers stroll through the park, they may encounter two types of situation. A standard passing by of another pedestrian or a sudden collision with someone else, which can cause stress. But why? In the book, The Hidden Dimension, Edward T. Hall postulated the existence of specific distances for different types of behavior. We can tolerate certain behavior at a particular distance from us, but the same behaviors would become completely unacceptable when they occur right next to us. Here we can see how a person appears from different distances. Imagine a situation like this. You are an unsuspecting potato strolling peacefully through the park, completely unaware that someone else might be just around the corner. You bump into each other and suddenly you have to make an immediate decision. Your heart starts pounding, blood rushes through your veins, and you quickly size up your opponent. One question echoes in your mind: fight, flee, or maybe talk. Time is ticking, and the advantage always goes to the one who takes that initiative. All this thinking, the adrenaline, the stress, it can really wear a person out. And all you wanted was a simple, peaceful walk. The repeated occurrence of situations like this causes the release of adrenaline and cortisol, which, unlike adrenaline, decreases much more slowly and can accumulate in the body, negatively affecting it. In a classic case of two people passing each other, there is a gradual process of overcoming successive personal distance zones, as they steadily move from the outer layers to the closer ones. There is time to properly assess the approaching pedestrian and appropriately react to their behavior. The problem arises in situations where, for various reasons, the outer layers are suddenly skipped. And here is the park we analyzed, Staszica Park, located in the northern part of Wrocław. As part of the data input into the simulation, we divided it into the previously mentioned obstacles and pathways. The characteristics of agents, as I said before, they have implemented visual field and interpersonal distances. And the results of our simulation. Here the fast presentation, if it could work, I hope. Yeah, something like that. As you can see, there are peaks in agent vigilance when he encounters other phantoms wandering around. I mean the blue ones. There would be another one in a short time. Oh, there it is. The red and the green one represents the number of people in specific human distances. The analysis of recreational areas through multi-agent simulations allows for significantly reducing the costs of redesigning faulty solutions, accelerating the design process, and organizing the best ones, as well as improving our lives by encouraging the use of parks and physical activity. In future works, we are going to clarify and standardize the Proxemic stress index. Thank you very much. Feel free to ask any question.
Okay, well, thank you again for the presentation. So do we have any questions? Not yet. Okay, well, regarding that proxemic stress index, do you have some kind of clue of how you're going to build it?
Right now, we have only the model we built as I presented it before. But yes, in the future, we would like to make more specific tool to use it, for example, when we need to score a lot of designs and choose the best ones.
Sorry. I didn't hear.
Hello, hey, can you hear me right now?
Yeah. It was just, yeah.
So yes, as I said before, we have only the model I presented before, but we would like to build specific tool to analyze a lot of designs in a short time.
Okay, right. We have a question from Patrick Thaïlandier who's asking, do you consider different profiles of people?
Yes, my supervisor tell me right now that we're going to do this. Right now, no, there are only one type of people.
Okay. If I might add, I think the proxemic, the different distance, how we perceive it is a bit cultural. Maybe I feel it a bit differently than someone in, let's say, North Africa or someone in East Asia.
Yes. in different cultures, there are other different distances. For example, in Argentina, there are the lowest in the whole world, and in Asia, they are much higher than in the, for example, Europe.
Okay, so we already have data on that. I don't know the topic, so
Yes, yes,
okay.
There are some data.
And do you plan on making that cultural? Do you plan on making cultural assumption, or do you plan on making a tool that is kind of universal? Or
Yes, yes. Even in today's model that we built, we can change the distances as we want.
okay, okay, very good. Well, Shimon, thank you for your presentation. We're a bit early, so that's perfect actually. And we're going to switch to the next one. Thanks again.
Thank you very much.
So next presentation will be simulation of talent retention and IT professionals turnover using Gama. Presenter may be Eduardo Ferreira.
Okay, and so I'm Eduardo Ferreira. I'm from Brazil, Rio de Janeiro. I'm a professor at Cefet. It's a university in Brazil, and my supervisor, my mentor, Oscar Rete, from UCES. It's a university in Argentina, in South America too, and it's a part of my doctoral thesis. It's a product of my doctoral thesis too. And the title is Simulation of Talent Retention and IT Professionals Turnover Using Gama. Okay, let's go. So what is turnover? What is talent retention? It's our object of research. And turnover, when you talk about staff turnover, it's a frequency which if people change job in the company. For example, when someone change your job, change your company, it's a turnover. You can measure this phenomenon with turnover rate. And there are a lot of authors, literature talk about in administration area, in a field research. And it's the most common, the most accepted concept and defined in term, it's a turnover rate, yeah. And the talent retention strategy is a HR strategy to retain employees in a company and minimize or control the turnover rates, because turnover rates is a problem, but in the little levels, it could be good to enterprise, and it make the organization more stability in a growth phase. When we implement talent retention strategy and suitable, yeah. And as I said, talent retention and turnover staff in IT sector is a complex and a big problem, because comparing the other sectors in the world, and it's a high levels in a turnover rate, in a staff turnover in IT area. It's about 15%. It's in the average level in the company, and in 80% in the Brazilian employees, for example, report difficulties to find talents they need to contract other people. So, and we show a little big numbers, and in the turnover and how much is important IT jobs, IT sector jobs in Brazil. And we have 2.02 million professionals in a technology IT professionals in a macro sector in Brazil. And a year in average level, we have more than 100,000 new IT jobs. So 4% are IT jobs in Brazil, but it's a micro sector in Brazil. It's in emerging countries, it's a similar case, for example, and in Argentina it's too, it's the same level, and other economies too. But it's not a problem just in Brazil. It's a problem all of the world. Other countries and happen this problem of turnover, IT staff turnover. So our objective in our article in presenting this in this Congress, and we made a simulation model to analyze how different retention policies affect job satisfaction and turnover rates. And we define IT professional, we name it employee. It's an agent in our Gama model, the species of agents in a Gama model. So our theoretical foundation are staff turnover theories. There are a lot of staff turnover theories in our literature, and multi-agent systems in a HR context, and others complex social simulation. So it's necessary a theoretical foundation so hard, so expensive to make possible we model a turnover staff model. And our methodology too, we using a Gama platform, and the first one will define our model specification. We define parameter selection, agent and environment configurations, and the last one, simulation execution and data analysis. So it's our IT staff turnover model. There are a lot of variables to make a decision to employee to decide leave or not our company, company. In the first one is the most important to decide. Job satisfaction, salary it's so important, no? And affinity of employing if your team or company, and the quality of with relationship between your boss, and perception of HR strategies, for example, policies retention. And there are make a coefficient beta to each variable, and it's so individual each people, yeah. And in the final, we calculate a P, a probability the employee leave the company. And in the final, in the summary of model, we have a global turnover rate model, and we can calculate the average level, the average probability to the employee leave your company. And it's a mix with a turnover rate tax. So our Gama beta model, we define a model staff turnover. We have a species employee. We define a grid to see the turnover rate and calculate the turnover rate. We have a experiment we name a staff turnover simulation. we define to make our experiments, our execution experiments, 3 scenarios. The first one is strong retention policies with a regular salary increases and promotions. It's a better scenario in the employee in an enterprise, for example. Great salary, promotions, good relationship with the others. And the second, moderate retention policies. And it's a more equilibrium in retention policies, because strong retention policies are expensive, no? And the third, weak retentions policies lacking significant salaries, increase of promotion opportunity. Yeah. So we have three scenarios: executions in a Gama platform, when our model implemented, and we define each cycle is a month, and when we define strong policies, the turnover rate is increases less. And we define weak policies, for example, the turnover rates grow up around the time, the cycles. And the employee satisfaction is opposite. It's a version of job satisfaction, and in our 3 scenario, the most strong policies reduce less around the cycles, and weak policies increases more. So our simulation, we use like a base of represent of prospects in a Gama. The balloon, the tutorial of balloon is the most famous model in Gama, and it's like our P probability of leave is like a stress in a people. When the most probability to leave the company, the size of balloon increase, grow up, when explode and left the company, you know. And we define the colors to represent the size and the level of stress of the employee and fee agents. Each cycle is an agent, for example. One. And our conclusion. And the Gama allows the creation of complex scenario with adjustable organization variables, and it could be practical insights for HR people, for HR manager. For example, my wife is a HR manager, and we introduced to our partners of the company. It could be a valuable data, but HR manager know nothing about Gama, for example. It's necessary people of IT, of experts in Gama, in other language, in other platform, to help or to support to make this analysis, for example. And our limitation is above future work tool. And you imagine, you design additional agents, for example, different roles of the agents, not in general employee, but employee boss, employee operational, employee tactical, employee manager. We could define different types of agents, employee, thinking BDI architecture, organization, to define roles, norms, for example. And we don't model agents interactions with each other. For example, it's important in the real life, in the real company. And we could be defining new variables using other literature, other research, and it's a great idea too. I think it's the most important, the comparison of results of organization real data to make it more realistic our design, our model. Okay. And our bibliography. Okay, thank you. Thanks so much. Questions? Finish.
Very much for your presentation, Eduardo. And I see we actually already have 2 questions. First, we've got Patrick asking, how did you define the parameters or variables of the model, all the beta 0, 1?
Okay. And let's, okay. To make a possible implement this model, we define if not a scenario desired, we use a random level. For example, it could be calibrate if a perception of the company to how much how approach, how behavior of their employees. Can you understand? It's calibrate with to make it executions. I use the random. So and make using scenarios. You use calibrating less or more defining the scenario. It's for example, we use a theoretical foundation, and in a research in an employee make variables to how much importance this variable is for the people, for example.
Okay, understood. Okay, we are low on time, but I will ask you the second question anyway, and we'll finish with this. Did your model enable you to understand new things about retention, or did it confirm or quantify things we already knew?
I guess it's more to quantify.
So, sorry?
To quantify.
To quantify.
I guess. Yeah.
Okay.
For example, I start with 100 employee. When you execute my simulation with my scenario, with my talent retention defined, and the final after of a year, I will be in the middle. I don't know, 15, for example, employee. Yeah.
Okay. Yeah, sure.
Okay.
Okay. Thank you again
so much
for your presentation, Eduardo. And now we'll go to the next one, which is social force model simulation of non-urgent risks from mass events in open public spaces with social group behavior.
So hello everyone, I'm Duarte Almeida. I'm a PhD student and invited professor at ISCTE in Lisbon, studying digital twins for tourism quality management, supervised by Professor Fernando Brito Abreu and Professor Inês Boavida, Portugal. I'm presenting a model that we developed, the social force model simulation for non-urgent egress from mass events in open public spaces with social group behavior. It's a big title. Okay, so the motivation. Many mass events occur in public open urban spaces. As we can see in the image, the celebration of a major football team from Portugal in a public urban event. Said events require risk analysis, scenario assessment, and adequate resource planning, such as security, emergency, and cleaning forces, during or after the event. Modeling and simulation is a valuable tool to test or to observe or hypothetical scenarios regarding these events. As such, we developed an agent-based model using Gama platform, obviously, to simulate non-urgent egress from mass events in urban public open spaces using a social force model extended with group behavior and social retention. This system describes the following characteristics: pedestrian behavior, environment geometry, exit choice behavior, crowd size and density, and social group dynamics. You can see here an example, an air view of a square that exists in Lisbon, the Praça do Município, and its simulated counterpart with the exits and with pedestrians simulating the event. The pedestrians traverse the environment and interact with each other using an extended version of the classic social force model, which is now implemented in the pedestrian model from Gama. In summary, in this model, the speed and direction of pedestrians are dictated by repulsion and attraction psychological social forces exerted by other pedestrians and the environment, as you can briefly see in the image. The classic social force model was extended with group behavior using a model presented by Saeed et al. In 2010, where pedestrians move in social groups such as family, friends, or co-workers, which walk together and maintain group cohesion. Usually at this type of events, of course, there are some people that go alone, but the majority of people go in groups, in social groups. Group members want to exit the event venue, choosing randomly weighted by exit activity. So we cannot predict as we can in an urgent simulation, in an emergency, we cannot predict where people go. So we model it by the activity of the exit, which means, for instance, if we have an exit that leads to public transportation or after party bars, will be more attractive than an exit that doesn't lead to those places. Before the groups start to move towards the end, they wait a random time simulating socialization at the venue after the event. Usually in this kind of events, people are not forced to leave the premises, so they socialize in groups or between other groups. We coined this phenomenon as social retention. These are the main parameters for the model: crowd size, group speed, group size, socialization time, the social retention distribution, exit activity, and the group social force model parameters. To validate this model and to extract model features to implement, we performed 3 focus groups with geography, architecture, and ergonomy researchers, and mimicked the monumental tour music and video mapping event, which happened in Praça do Município, as you can see in the figure. We used cell phone count data gathered during the event and provided by the LX Data Lab initiative that provides data to provide solutions for the city, for Lisbon. We then ran 1,000 simulations for controlling the stochasticity of the model. We published an early version of this model in my master's thesis in 2021. And recently we published the final model in the journal Simulation Modeling, Practice and Theory. Now I'm showing you brief demonstrations of the model. In this demonstration, we can see a time lapse. It's a time lapse of people leaving the event without social retention. So they leave all at the same time. It's very brief. And then we can see an in close demonstration of the group cohesion. The pedestrians in white represent groups that are socializing. Hence, they are almost static. They only move so that they can give way for the groups that want to leave. This is in real simulation time from my computer, which is not very good. So the performance is not very good also. You can see here a more open view. Some groups are starting to move, as you can see. Okay, I would like to acknowledge Dr. Patrick Taillandier for helping me during my master's degree to learn some intricacies of the tool, and Dr. Arnaud Grignard for inviting for the presentation. And now I'm done. If you have any question, feel free to ask.
Yes, thank you for your presentation again. And it seems like we actually already have one question from Luca, which is asking, how long does a total simulation take to execute? Are you trying to expand the scale of your model to match more closely the number of real person in the area?
That's a tricky question because the way we implemented our model, it has some performance issues. We did some performance tests, and it's hard to scale this with with our implementation. It's hard to scale with more people. The people that we tested in this scenario was from the cell phone counts that we gathered during the event using the cell phone counts. Maybe it's not accurate, but it's what we could use. But yeah, it needs some performance scaling. It needs to be, or the algorithm should be better done, or using other types of parallelization, and that's it.
Okay, well, turns out Luca is working on how to optimize simulation by splitting them spatially. So maybe he can contact you or something if it's something that interests you. Okay, you kind of already answered that one, but Frank was asking, have you calibrated your model on real data? So
yes, from that's cell phone count data.
Yeah. We have another question from Manon Prédumeau asking, have you considered various social relations in groups, family groups with children, friends group, couples? It might impact the cohesion in the group and the distance pedestrian maintain in the group.
That's a pretty good question. I think I'm not really an expert in group cohesion, but from what I perceive, yes, you can have different cohesion forces, because in a family, you want to, if you have your kids or your wives, whatever, you want to hold hands with them and you want them really close to each other. Hence, if you are looking for a group of friends, they can be more scattered. For now, this model does not account for that, but it's an interesting point.
Okay, sure. We have Patrick Taillandier, which you just cited, that is asking, are the behaviors very different in a crisis context?
They should be quite different, because in an emergency scenario, people have panic. They want to leave the premises as soon as possible. They are not choosing where they go. They go where they can. So if they will choose an exit, they will choose an exit nearest to them, or the least clogged one. So it should be a balance. And group cohesion, it's different. So maybe in the panic, people maybe run from the use their own. They scatter. With the panic, they maybe lose the group cohesion. So it's it's different. So that's why we made this non-urgent, because there are a lot of models regarding your urgent evacuation, but we focused on the non-urgent that we did not find a lot in the literature. Yeah.
Okay, thank you. I think that's all for the question. I might add that unfortunately we had Professor Benoit Godou that just left, but he worked on something very, very similar to you, the same kind of evacuation from a place in a French city. And he found out that the model you're using, which is the social force, it works well when the density is not that high, but when there is a high density, it doesn't really fit the data. Is it something you've noticed, and are you something you're trying to work on, or?
I did not account for that, yes, but I've heard, I've seen some studies that talked about the efficacy of social force model, but yes, maybe there are better models to model that kind of simulation. Yes.
Well, if something you're interested in, I think the solution they provided together with his PhD student was to have the social force for low density, and when the density is too high, they use some kind of reinforcement learning. I don't know what kind of algorithm specialized that was trained on real data for high densities.
Yeah, interesting, yes.
Okay. I think we're good for the questions. We are on time. That's pretty good. Okay, perfect. Duarte, thanks again for your presentation. And
thank you.
We'll go to the next one. So I guess this time is Mikołaj Szurlej, Strolling Activity Simulator for recreational audits, measuring the health impact of the built environment.
First of all, thank you very much for the invitation for this conference and for coupling my team with our presentations to biological group. And of course, we are doing anthropological models, we are doing psychological, but still we are inside of biology, inside of human biology. And we use generally, like mentioned before, Hamilton rules and Dunbar numbers, it is still game theory built inside of biology. And those very simple models of human and animal behave, we are trying to implement in our models. Of course, differentially developed, and now I will try to present our best and most developed model we are using for taking services for our city, 1 million Wrocław in Poland. And we are in still collaboration with local government to check before building new gardens, new public parks, checking what is recreational, possible, of course, recreational efficiency for particular garden, particular public park. Of course, main problem is lack of physical activity. We know that physical inactivity is European or first world pandemics. Most of civilizational sicknesses are joined with this epidemics of physical inactivity. And the problem is how we could build our cities healthier to make in our citizens habits, how to change behavior, which inactive is harmful for people, how to activate physically without any orders, without any strict rules. We can only suggest, we can only build some environments which could change their behavior, which could change learning. And this way of learning is absolutely different than in large language models. We are trying to mimic biological process of getting habits. It is absolutely different than logic learning. Getting habits, it's like changing skills, changing behaviors, and it works in the side of our brain, but in very old brain, in this very basic part of brain we have a common, for example, with reptiles and even birds. So it is very primitive, very original way of having possibilities to change behavior. And all the model, all the scenario is, if we are sitting home and occasionally we go outside to get some activity and we'll have just not nice environment, we are not motivated. If we have something motivated, if we have something nice, probably we will prolong our strolling, our walk, and this time of strolling will increase a little bit. And we modeled this small increasing like part of very differently understood memory, like motivation. If we spend more time in our strolling, in walking in green areas, we are next day a little bit more motivated to go outside and be open for individual experience. And in this model, every agent learns only from his own experience. And of course, the way of using this, how our main simulation works, we have, let's imagine we have architectural competition for some big garden, some big park. And let's say we have in the finals 20 of very good designs. And what we are doing for every, we put every particular design from finals, every of this 20, we put inside of Gama. Of course, we change to layers like in Gama with pubs, with buildings, and we populate it in virtual agents as close to census, to demographic data we have from outside. We, of course, try to calibrate it to the situation, how often people use this greenery, how often they walk around. And because it is our city, we generally know how often this unbuilt park, this unbuilt garden is used now. And it is calibrated. So state zero is calibrated to existing behavioral state, not only physical, but behavioral state. And when our model is calibrated, we put one by one those design, those layout proposed in final of architectural competitions, and we produce using this simulation in Gama, of course, this chart of we see on the right hand side, we have activity of people. And in this blue line is how often, what percentage of people are walking half an hour per day, what is in health sciences, this level of behavioral safety. If we are active half hour per day, we are safe statistically. So it means it is very probably we will be will live healthy life till we are 80 years old. So it is what we could safetiness of environment. So and in left hand side we have also, it is kind of charter. In our model, Gama will put colors. Green means the lighter green means more people used this green, walk through this greenery. In buildings, red color means 80% of people, of inhabitants, are active on this level, about or above half-hour pay per day. So in this simulation, we could estimate for every particular design from this final of architectural competitions, this factor, and we can do clear ranking, which design is the best for health of inhabitants. And as we see, those colored lines, they put direct ranking. In every simulation goes through one virtual year, and simulation is done for about, as I remember, 16,000 of human agents, and every decision to go from, if I'm in house to if I'm home, to go walk, or if I'm during the walking, is it nice and I will continue, or is it not nice so I will go back home? This decision is done by in every virtual minute per 4 or 15,000 people per year. So this quantity of those virtual decisions is quite big. And during doctoral studies for my friend Mikołaj Szurlej, he was checking a lot of Chinese gardens, Chinese public parks, and this quite simple model in Gama was quite efficient in prognosis of activity. In gardens, we knew from Chinese data how often people walk around there. So maybe this model itself seems very simple. It was quite efficient and produced very stable and extremely stable outputs. With many changes of perimeters, it was still very resilient for changes of the ranking itself. And what we did during this simulation, we proposed the one perimeter called PIRI. It is Physical Inactivity Reduction Index. So it is the one number which produce the ranking which propose of design is the healthier for local community. And during Gama simulation, from the layout during the simulation is converted to one number, and in the value of this number we can do objective ranking which garden is the healthier. And this physical inactivity reduction index means exactly the difference. We know how many people in this environment, in this neighborhood, walks enough, so walks at least half hour per day. And if the design proposal will change this number, will increase the group of active people, it means this pathological physical inactivity will be reduced. But our Gama simulator could answer us how much it is reduced. So not really. So for landscape architects, of course, they for today use the paradigm more chlorophyll, more greenery is always the best. But our simulator shows not only greenery, but greenery connected to human, to dwelling, to housing. And this value of greenery is estimated or measured in the level of physical activity. So it is not only greenery as value itself, but we can simulate value of human health produced by landscape architects. And for now, we are open contracts, and our students during their
is everything all right? We lost the sound. Maybe if you have a technical problem, just write in the chat so we know. Hello again. I think you've been you lost your connection maybe or something.
Yeah, we lost the connection with output device. Can you hear me now?
Yeah, we hear you all clear.
Okay, thank you very much. So I will switch that headphones.
Okay, so now can you hear me?
Yeah, we can hear
okay, you. And how long?
Around 1 minute.
Around 1 minute.
Yeah.
So probably anyhow, I should go to the end. And this presentation, we built the model of human activity, including learning. Okay, we have some technical problem again, but still, can you hear me?
Yes, we can hear you, no problem.
Okay, so we built the model of based on reinforced learning, but in the very basic Skinner's model of learning. So it is positive enhancement, positive impact of positive activity. If something is nice, it motivates us to repeat this action. And if this action is more and is increasingly gathered, increasingly understood by like a nice, it could change behavior in very long time. It could change everyday habits. And it is what was our goal, to build the machine of virtual agents which could get habits like people and use them to experiment in architecture to reduce quantity of experimenting on living people. And for now, it's all. I hope I was still online. I have problem with my camera. I'm not visible as I see. Okay, so for now it was all. And could you, if you have any question, I'm very open to answer it, please.
Okay, well, thank you for the presentation. Okay, unfortunately, some technical problem, but it happens. I see that we have one question already. How did you use the concept of affordance in your model? From Elisa Coy.
The model of affordance, so what environment afford us to do? It is the model from theory of affordance was in the ecological approach to visual perception, as I remember. It was the title of the book, and we used it very directly. So it is what is the possibility understood by agent. So it is not passively any feature of object itself, but how this object is able to be perceived, is able to be used, is able to be remembered by particular age, particular animal, particular person. And so we used this model very directly from biology with no, maybe we did some mistakes, but it was not our intention to, to misunderstand biology. We still believe evolutionary game theory is still very open, and we try to use those models in safety studies, in health studies, and also we are now started to build neuroatypical agents, and we are quite advanced to model autism and Asperger spectrum. And in next presentation, we showed our approach to ADHD neuroatypical agents.
Okay, thank you. So we're running out of time. I had question on how you did the agent exactly, but we'll skip that. And maybe I can just ask you, so it seems like we can use your model for some kind of city planning, let's say, something like this. Did you consider making it a tool distributed for that? Maybe quickly,
now,
sorry.
We are not computer scientists, and we wrote Gama code as we were able to do it the best. But of course, it should be checked by you, by specialists, and we are very open to clarify it and open it and to promote those paradigm of biology inside the city planning. And health and safety problems could be resolved as, for example, non-go areas around of Paris, and this territorial conflicts, how it works in contemporary cities. It is what we would love to do, but for our biological models are still, the ceiling is to have enough time and quality and skills to write proper Gama code. So we have plenty of graphs, plenty of models itself, but we are very weak to code it, and this recreational audit was the best we were able to do.
Okay, well, thank you again for your presentation. Unfortunately, I have to stop it right now.
Okay, thank you very much.
If you're interested about the city planning parts, maybe you can check the planning, the program of the Gama Days from yesterday. We had quite a lot of people talking about it. And now for the next participant, which is presentation is towards architectural support for cognitive behavioral therapy for ADHD, modeling and simulation of daily environmental interactions for mental health enhancement. Maybe Bernard.
Okay, so good evening, everyone, and me and my supervisor, Maciej Szarejko, would like to present our idea, our proposed application to agent-based modeling ADHD DMA. And okay, in this presentation, we work. Our work combines BDI and cognitive behavioral therapy principles to create. Our aim is to create supportive environments for people with ADHD. Here we have that's basic model of this classic BDI model, Gama, and when we switch it horizontally, we would like to present on this scan how it works to apply here the multitasking and the way of thinking of working ADHD. So here we have this the basic model using BDI, where we have a lot of stimulus from environment, but most of that is blocked by barriers or cognition focus. And here I added that bold line to underline that working memory, which is very important here. On the next slide, we will see why. Here, I present that multitasking scheme. It's not so easy, but still readable. And how it works for neurotypical person. On the other side, we have here we have multitasking scam, but with ADHD problems, which makes, for example, task switching difficulties, environmental overstimulation, or executive function challenges. And it makes harder when we have, we can observe here the I called it cumulus, but it's a lot of stimulus which together makes a big problem for definition of working memory. And at the end, the ADHD person see something like this during their daily exercises. And here on this slide, we see our proposition to how it works when for example, this cognitive behavioral therapy is turning on. The therapists try to therapist person presence also helping to lead that execution plans to the end, or come back to the others which are started before. And here that's presence of therapists. We change to trackers, environmental trackers, which would be very helpful in and can lead to support that executive plans to the end. These trackers. I called here examples for these trackers. It could be something simple like alarm clock or reminder, but if we talk about architecture and or special engineering, we can use this physical objects to help person to keep focus on that plans, or helping to not overlapping that plans or thoughts or desires. And thank you all for attention. We can switch our output signal to un momento. Tak. Okay. Okay. We are open to cooperation and respond to your questions, but we have problem with this hardware input now, and we can apply that responses on here, maybe chat or mail. Yeah, mail. Is it possible? Because here I am only hearing one. Yeah. Have you maybe any questions on the room?
Yes, sorry. I thought you were having an issue in discussing between you. Sorry. Okay. So do we have any questions yet? Okay, not yet. From what I see, it's pretty exploratory. So I don't know, but from what I've seen, there for the moment there is no model yet. You've mentioned the BDI.
Yeah.
And so there is a module for that in Gama, of course. Have you tried playing with it a bit? And I'm not sure to understand how you want to apply this.
I understand. Now we could focus on that planning of schemes to later apply that for Gama. Now we don't have some code or Gama simulations to presentation, but in near future. Yeah, we are working on this.
Okay, yes, sure. But do you have a rough idea of what would be the dynamics basically? Because here, okay, you've got the cognitive model, but what would you do to make it interesting, sorry, as a model, as a simulation?
Interesting as a model.
Yeah.
How it we will make it interesting in simulation. Yeah, can I understand?
Yes, correct.
Okay. I think interesting is it would be interesting to observe that agents, individuals with this different model of behavior. And I don't know, maybe some
maybe a case study where it will showcase the difference properly. I think it's often what we do with simulation. We use that to showcase.
Excuse me, I don't know if I understand well. Could you paraphrase or repeat?
Yes, so you've got that model, pre-model of the ADHD behavior, right?
Yeah.
And what I think is it could be interesting to showcase how it changed the actual results in some kind of context. Maybe that's my point of view, but yeah.
Yeah. That's my too, but it's still in the near future to plan to present that on this simulation. It will be cool to observe that what we achieved here during our paperwork. And if you think when would be the possibility to present something like this, maybe next year, but it would be very cool to make it as fast as possible.
Yeah, sure. I think as soon as you have you've done the work on the model theoretical, so I think the simulation part is not that hard to do. I think it can be achieved pretty quickly. What is often hard is to find a case study that really showcase the difference. Yeah.
These researchers with statistics and so on. We would like to do that, and as architect, we need to check that behavior models in our surroundings and architectural surrounding. We will work on this. Yes.
Okay. Yeah, understood. Okay. I don't see more questions, so I think we can go to the next presentation. Thank you again for this presentation.
Thank you very much. Sorry for that problems.
Yeah, it's okay. No problem.
Thanks.
Next, we have a presentation about multi-agent modeling to assess robust emerging properties of biological systems from Daniele Proverbio, I guess.
Oh, good. Thank you very much for organizing this. It's been very fascinating so far. I hope to contribute with my 2 cents on this roster of presentations. These also will be slightly different from what we heard before, so there will be a little bit of preliminaries before delving into the results. But overall, this talk regards the use of multi-agent systems to, as you can see in the title, assess robust emerging properties with a focus on microbiological systems in particular. It's been a work, pretty collaborative one. Part of it was done at the University of Turin and then conducted at the University of Trento. As mentioned before delving into the results, I'd like to give you a couple of definitions. So regarding computational robustness, so in general, we define it as the ability of computing systems to maintain functions despite alterations of individual programs. And this pairs the notion of dynamical robustness in dynamical systems, which is basically the same thing, by considering uncertainties on parameters or dynamical noise. Emerging robustness, what we mean by that? Well, we all know about emerging properties, so properties that are showcased by a system that were not encoded into their components. And here we're interested in looking at what's the minimal program out of a set of programs P's, such that the minimal length of the set can anyway ensure the emergence of the properties of interest. I don't really need to read the definition of multi-agent systems since this is exactly the perfect conference to do it. But it might be particularly relevant to stress that for certain scopes, microbiological systems in particular can be aptly modeled as multi-agent systems computing machines. And therefore, we can apply the above definitions for studying those types of systems. As biology-inspired case studies, I will mostly focus on this guy here, which is Dictyostelium discoideum. It's a social amoeba, basically a collection of cells that eventually manage to gather, performing the so-called decentralized gathering task. And I'll touch upon new results about the network formation in Physarum polycephalum, which is a slime mold. Now, the goal of this work is to first develop a multi-agent systems model to reproduce the emerging behaviors of both systems, and then assess their robustness and minimum models. Since you're also interested in the technical details, I'd like also to mention that the choice of Gama was primarily driven by the language and its easy to use properties. Since for biology, there are a couple of additional tools like Morpheus, which were initially tailored to biological systems, but eventually they are slightly less flexible. So I'd like to congratulate the developers of Gama for providing such a nice platform. All right, now getting into the main protagonists of the study, Dictyostelium discoideum, as mentioned. Can you also see my pointer, which would be nice to point a few things on the slide? All right, cool. So it's a social amoeba in that it has a multi-stage life cycle. It starts as single individual cells, which then can aggregate if they are starving, basically if food is depleted into the environment. And then by partially fusing their membranes, they're able to develop a multicellular single organism, which eventually can create stalks and produce spores to send into the environment and so on and so forth. So in biology, it's particularly useful as a model organism since it goes from a single colony of multiple cells to a single multicellular organism. But right now, I'll mostly concentrate on the two phases, so the amoeboid and aggregation phase. And this is actually how we can map these biological system into a multi-agent systems model. So single agents will be, single cells will be modeled as our agents, which possess an internal architecture that allow them to dynamically interact with the environment. And all those processes are informed by biological knowledge. I'll show you later also how can we set the parameters. The way they communicate with one another in the real biological setting is through sending cyclic adenosine monophosphate molecules. And in this case, these will also be rather stupid and reactive agents, but nonetheless agents, which is a trick that helps to speed up the computational simulations. And we can also start playing with the environment as a dynamical grid, and also placing on top a few like obstacles, mazes, and the like in order first to reproduce what's been observed, and second to try and use this as a test bed to produce new observations for experiments. This is basically how things are implemented. So in orange, you can see the environment. The green dots are actually the amoeba agents, and the blue dots are the molecules. We're not really interested in how they look like. This is just as a way of navigating through the simulations. As you can see, the flowchart of decision-making for the amoebas is fairly complicated, but not that much. I mean, they're still single cells. The environment can be dynamical in such that each cell of the environment can change according to some probability, and those cells represent food. So it's basically in the flow chart, it's here. So if there is food, then there will be in one state. If there is no food, the amoebas will be in another state. So by changing the probabilities here, we can tune the states of the amoeba. Another source of uncertainty is the probability of misprocess an incoming signal that we call PA of agitation. And a third source of uncertainties is the presence of obstacles, because those can trigger certain methods, certain functions to explicitly look for obstacles and avoid the obstacles. And this was particularly designed because it's still unknown, actually, whether those cells do possess those types of obstacle checking routines, or if they manage to perform obstacle avoidance by other means. So by including this requirement into this computational testbed, we can actually check whether they would make sense or not in order to produce the emergence of decentralized gathering. So these are two simulations. Well, both are sped up quite heavily so that I can fit into a 15 minutes presentation. But in general, you can see that the decentralized gathering process occurs. So scattered agents start to coalesce into a single center. And in the rightmost simulation here, you can also see that they manage to circumvent obstacles and eventually gather into a single cluster. We also define quantitative metrics in order to look for the whole process in a quantitative manner. For the homogeneous environment settings, the variance is pretty straightforward as a metrics to measure dispersion of the colony. Whereas for the other case, we had to look for like a local metrics, like a clustering coefficient. So basically looking how many neighbors are close to each cell and then averaging over the whole colony, since variance is only defined for Euclidean spaces. And then we can basically get all our results. Here we can see that the variance can depend upon the number and the density of agents, up to numbers and densities which are actually compatible with the real number of cells in biological colonies. And we can also see that they manage to cope pretty well with the environment, with the addition of environmental noise, since the time that it takes to perform the decentralized gathering only scales up linearly, which was not trivial. Like it could break down much more heavily. Instead, for what concerns the agitation probability, you can see that this time to get into the cluster scales up exponentially, but this is halfway through, because actually what happens in biology is that the agitation probability goes up to these types of numbers where the scaling is still rather linear. So even in this case, the colony as a whole is pretty much able to cope with uncertainties and perform the decentralized gathering task. And finally, also we can check for all the other parameters, for instance, the shooting time, so the relay period by which those cells send pulses of chemicals into the environment. And in this case also, the time scales linearly with a fairly low coefficient. So overall, the colony is pretty good. In addition, we had tested that hypothesis about what happens when obstacles are present. And in here, you can actually see upon several different investigations. So one just using the obstacles, like one single obstacle, then inputting multiple obstacles, and finally also adding uncertainties on top of obstacles, that eventually the colony, with or without P1 and P2, corresponds to having the explicit sensing for the obstacles, and P2 corresponds to not having it. They basically showcase the same performance, which actually tells us that in principle, biologists never found those mechanisms because maybe they are not necessary. Because it's by sending the chemicals which are absorbed by the obstacles that the whole colony manages to perform obstacle avoidance and eventually decentralized gathering also in heterogeneous environments, which is also pretty cool. Unfortunately, we are theoretical and computational people. We don't have a lab, so we cannot really test this hypothesis, but it's now published, so any experimentalist can actually delve into those questions. And finally, since I see that I'm kind of running slightly late, I'd like to mention that the same process can be applied to other types of systems. For instance, the slime mold question, in which we can actually input several biologically related parameters, test both what happens when we change them deterministically, so akin to sensitivity analysis, or when we allow them to span within an uncertainty interval, which is robustness analysis. And those guys are actually able to perform network formation in this case. So basically linking two food sources, depicted here as the orange dots, by creating a single link throughout them, which we can also see is pretty well close to optimal. So there's a high success rate in doing this, and we can also measure that those links are as short as possible, which also corresponds to what's observed in biology. So to wrap up, I showed you the power of using gamma and agent-based simulations in order to assess the emergence of properties in microbiological systems, which is something that analytically we cannot use. And with cellular automata, for instance, I mean, those are too simplistic to actually capture the full characteristics of even biological cells. Thanks to those simulations, we can actually assess the persistence at macro scale of some properties given the micro scale uncertainties, which is also something particularly cool that we can only do with agent-based simulations. And finally, to use the whole machinery to guide by inspired design of artificial self-organizing and robust systems. This is kind of work to do. So far, we have concentrated on the analysis, but synthesis can also be possible. Finally, I'd like to mention that I'm also guest editing a special issue on robustness and resilience of systems and networks. So if you'd like to have a look, there's the QR code here. So yeah, thanks for listening. I'm open to any question.
Well, thanks a lot. Once again, very different from the others, but so refreshing, very interesting. And it's nice to see that emerging properties. I mean, that's why we love the multi-agent. So now for the question, I see we have one from Frank already. Apart from the ease of the language, have you found any feature in Gama that are or would be useful for your case study?
That's a nice question. Well, surely, well, on top of the syntax, the ease of implementing motion, that was particularly welcomed, since at least we didn't have to care about like parallel motion or whatnot and scheduling and stuff. That was very, very useful. Well, and then there's always the thing that you kind of stick to the language that maybe you knew from other projects that was particularly useful. Because on top of microbiological systems, I also had a project on macrofauna with a colleague from ecology. And to model the decision-making of big animals, we were using the BDI framework, for which Gama was particularly well suited. So adding one on top of the other, eventually we got like fairly proficient with Gama, so we kind of stick to that. Maybe what can be particularly useful, maybe for the future, could be the possibility of like merging and fusing agents. So instead of like deleting one and making another one bigger, like making a full fusion, that could help in proceeding from the decentralized gathering to the stalk and slug of the amoebas. But yeah, there are maybe also other workarounds for that.
There are ways to do things similar to this in Gama. You have ways to integrate agents into agents, so maybe that's something you can look up.
Yeah, halfway.
Maybe, maybe not. And there are also some work on what's called multi-level. We talk a little bit in the beginning. Sorry, we're running out of time, so I'm not delving into that. I'm just giving you your last question. So from Jean-Daniel Zucker, who is asking, thank you for this interesting talk. How to scale up the number of cells if one cell is equal to one agent, and couldn't you consider agents as clusters of cells? You might have done it already. Oh, yeah. Okay.
Yeah, absolutely. So considering agents as clusters of cells can be done up to a certain extent. First, because of the technology that we just mentioned, but second, also regarding the biological interpretation. So it's like a bit tricky. What we did instead was to actually perform a study about when the system becomes scale-free. If there's a point such that augmenting the number of cells doesn't change dramatically the qualitative and quantitative behavior of the system itself. And we did it, and thanks to the properties of our system, we found this threshold up to a number that was like about roughly 3,000 agents. So thanks to this study, we managed to fix the simulations to 3,000 to 2,500. That anyway took reasonable time to run, and we knew that any addition in number of agents wouldn't have changed things dramatically. So we resorted to this trick.
Okay, sure. Well, Daniele, thanks again for your very interesting presentation. And now we're going to go with the last one. I'm sorry, guys, if we're a bit late. We kind of knew it was going to take a bit more time than planned, trying to stick to our 15 minutes. So last presentation is using Gama to run reinforcement learning simulations of disease spread, including the effect of pathogen fitness. From Andres Colubri, I guess. Yes, I see you in the chat.
This will turn out to be more of an exploratory presentation about the exploration of this idea, not necessarily the implementation yet. So I'll go right into it. Let me hide this and slideshow. Start from the start. So as you mentioned, the title of this presentation is Using Gama to Run Reinforcement Learning Simulations of Disease Spread, including the effect of pathogen fitness. I added this planning on using Gama because this is what we intend to do. This is, I would say, a continuation of work that I started doing with Gama and more specifically with ComoKit back, from the early days of the pandemic. And I think there's a lot of potential to combine agent-based simulations in a software such as Gama with experimental data. And hopefully, even though I won't be able to show results from the Gama, hopefully I will bring some ideas that are interesting for the community and maybe, this leads to some discussions. So I'm based at the University of Massachusetts Medical School. And yes, I have my lab that focuses on infectious disease research, basically modeling. And I think I will start by talking about a little bit kind of this context of this project, which is to use games to learn about infectious diseases. And we can start, the idea of playing mock outbreaks is not new. Many of you are probably familiar with different types of outbreak games. Like here, I'm showing this famous board pandemic game. But as it turns out, we can also use games, outbreak games, to learn about real epidemic processes. This is an example from not that long ago. This is 2007, between 2007 and 2012, a group at the University of Cambridge, they developed a mobile app, but for phones that prior to the iPhones. Sorry, I heard something. I don't know if was the other question. Okay. Where the idea was to detect proximal contacts between individuals using this app, and then using that information to reconstruct contact networks, and using those networks, trying to, to understand properties of those networks and how they affect epidemic processes. So the key thing here is that human behavior is critical to understand disease spread, as we probably all know from the experience during COVID, but that's hard to model accurately, right? And an observation that is not new, a new observation is that if we design these virtual outbreaks carefully enough to try to answer public health or epidemiological questions in epidemiological studies, maybe we can then incorporate somehow unprogrammed human behavior, real human behavior into these models by doing some sort of let's call it real life simulation or experimental game. And this kind of goes back to, that's actually from a quote from a publication from 2007, where the authors were epidemiologists were looking at this interesting instance of a virtual outbreak that actually was essentially where the participants were human individuals were playing this World of Warcraft, a massive multiplayer game. And what happened is that there was some sort of unintended viral spread. Maybe some of you have heard about it because it's kind of famous. And the realization was, oh, we can actually see behaviors here that really match real behaviors when there is an actual pathogen in real life. And so we could try to understand things by looking at these virtual or kind of gamified outbreaks. And that's the idea that has inspired the project that I'll talk about. And of course, I want to connect this with Gama, but first I need to introduce a little bit this project called Epidemica, which is a platform for this type of open world epidemic experiments where people install an app, and this app has a number of gamified options to, for example, quarantine yourself or acquire different types of protections. And that data is collected during the game and can be used in different ways. This is some results where you can see some picture of the people using the app, some screens of the app, and network data collected during a particular simulation. And as I mentioned, this smartphone app tries to gamify these kind of decisions through doing an outbreak, during a kind of outbreak game. We have created this type of kind of gamified interface with characters. Oh, I think I lost a couple of things there when I copied and pasted, but essentially this simulates transmission through Bluetooth and also disease states. And it transmits that information using this kind of game-like interface. The app also includes other features like quizzes that you can use to gain points and a whole array of different features that I don't have time to talk about. But all of those decisions that people make through the game can be then utilized and see how they correlate with, for example, the spread of the virtual disease. And I think, well, this is the reason why I was coming back from the UK, because I was there working with my collaborators who are working on these, who are developing these technologies for proximity sensing. And this is a group that early in the pandemic, they proposed the use of digital contact tracing, essentially contact tracing through smartphone apps. And essentially, well, just to give you a little history, this Epidemica platform is based on a project called Operation Outbreak, which is a platform where we run the simulations already for several years, mainly with a purpose, the aim of education. Essentially, we want to run these games to educate middle and high school students about how epidemics spread in the population and what they can do. They can play roles, etc., etc. But the idea is that we are taking this Operation Outbreak into a direction that is more focused on the research, on kind of this behavioral research. And the data that we can get from these simulations or these games is quite realistic. Actually, it's just comparing showing some kind of plots of cases from these games. And we have this using many settings. Just some pictures here of different schools, colleges, conferences, etc. And in a way, a little bit some food for thought is that we could think of these other games. They're kind of Asian-based simulations, but where the Asians are real people, right? And they take place in real life. I mean, it's kind of obvious, but I think it's an interesting thought because this is kind of, in a way, what connects with Asian-based models. Yes, we have real agents, and then we'll try to parameterize kind of the kind of where I'm trying to aim to is that can we observe behavioral properties from the participants of these epidemic games in real life? Can we then try to parameterize the way agents play in an Asian-based simulation, right? So, and let's put it this, I'm gonna kind of cover ground quickly here. There is a lot of theory about how human behavior can be modeled mathematically. One of those theories is called prospect theory, which another one is evolutionary game theory. And I bring prospect theory first because it's a very powerful framework to model human behavior, because it allows us to, it considers that, it assumes that people don't behave rationally. They are bounded rational. So they don't take the optimal choice, right? That's very important, especially when people are faced with uncertain outcomes. So there's a whole, again, there's no time to go into this, but you can easily find the information online about how prospect theory models the subjective perception probability, which is essentially the plot on the left, which is essentially you have the identity, the diagonal would represent the objective probability, and this kind of different blue and pink curve will represent the subjective perception probability, where maybe individuals overestimate or underestimate extremes of those probabilities. So this theory is actually it's not sufficient for what we want to do, because prospect theory is a static decision theory. There's no change over time. It doesn't consider learning. So what we want to do is to have the possibility of agents adapting to changes in this environment. The environment in our case is the outbreak that is changing over time. Here's where we connect this prospect theory with evolutionary game theory that allow us to mathematically study how in populations the behavior dynamics capture how things tend to happen in the long term, to try to maximize some payoff following, in this case, here is a matrix showing kind of this idea of cooperation or not. If you're familiar with game theory, this is a payoff matrix that you might think of in the case of all these type of game theory games, right? Like the prisoner game or the prisoner dilemma game, etc. So we have a way of combining these things in the context of outbreaks. And what we do this by defining payoff matrix where we have a payoff matrix. We have a payoff matrix where what happens is that there are rewards and penalties based on certain decisions that people do during the game. Now, if you think, okay, people are playing this game with the app and they are actually making contacts in real life, and they decide if they want to adopt certain protective measures or not, all of those decisions have points associated to them. So we can actually construct an explicit payoff matrix. And with that matrix, we can then write down, say, if we have a SEIR type of epidemic, then we can write the equations. Here we can apply the evolutionary game theory that allows us to write through this replicator dynamic equation. Allows to combine the EIR dynamics with the evolutionary dynamics due to the kind of the payoffs. And this is where things kind of come together, because here what we have is this equation that we can solve. And in this equation, I won't go into the details. There are some elements here like this W, for example, and this V, which are actually the functions from the prospect theory, which are those plots that I showed before that quantify or model the subjective perception of risk. And that's how things come together. In this equation, we know everything else because in our epidemic process, all the parameters except the behavioral parameters of the prospect theory model are known because we define how the pathogen spread. We define everything, essentially. So the question is, this kind of exploratory question is, can we do this type of reinforcement learning simulations in Gama to simulate bounded rational agents? And the question is that this is the kind of the traditional framework for reinforcement learning. You have this reward function that you can express in terms of the Bellman equation. And here you have the probability of taking the probability P of taking a state, going to a new state from some previous state with a certain action. What is the immediate reward? There's a discount factor associated to that. So this is kind of the standard way of implementing reinforcement learning. Well, apologies for the sound. It's just that I'm in the baggage claim area. So, and what we need to do is actually to use a modified version of that, where instead of having just a straight probability, we have to modify them by the functions that incorporate the subjective perception of risk. which will be something like this. The thing is that all the methods that are known to solve this in a standard reinforcement learning simulation would not apply directly, because now we have these nonlinear transformations. So that's kind of the problem here. We need to find a way of doing reinforcement learning with these kind of bounded rational agents, which we are modeling with prospect theory. So I think that's kind of the challenge we would like to do. And I think I started finding how to do as well, obviously, because I did work before with Gama. I was thinking how we can do this in Gama. And I found some, I was searching online. And actually, there's some examples from, for example, in the Gama community forum about how to implement Q-learning, which is essentially another terminology for that. So I need to explore this a little bit. This seems to be a very basic example. So I will need to see how this could be made into what we need for our epidemic games. There's a paper that seems to be quite new. It's from last year, and it uses Gama to do reinforcement learning. But these are all, I believe, using the standard reinforcement learning, essentially no prospect theory to incorporate bounded rationality of the agents. So this is something kind of that we need to explore. Now I'm gonna end here because I know I'm the last one, and you know things are running late, and there's a whole other component to this, which would be to incorporate evolving pathogens as well. We have agents that have adaptation or adaptive behavior. There's a whole component to this that would consist in making the pathogen also changing over time. I mean, maybe responding to how the agents change their behavior, but that's too much. I think I gave, and hopefully I provided enough, you know, materials or ideas that might stimulate, you know, discussion, hopefully. And yeah, maybe I can talk about this next year with more time. So yes, I think kind of this is also a call for, not for help. I mean, this is a little bit of kind of, this something is kind of what I want to put here is a call for, well, it's something that people might be interested in working on together, or you know, how to actually do this type of prospect theory-based models in a nation-based simulation. So if people are interested, and specifically how to do it with Gama, right? You can reach me with email. And yeah, that's it. Thank you so much.
Thank you, Andres, for your presentation, and it was also once again very different from the others, still interesting. So I don't know if we have questions yet, but turns out I participated in the work you cited in the event, so I can maybe give you some insights.
Okay.
The points you had about maybe the prospect theory, right,
Yes.
is not exactly adapted to what has been done. I don't think it's the case, because we have been doing something that is very generic. Basically, we've done a reinforcement learning loop, and we can exchange any kind of data. So if I understood correctly, you just have a few more parameters, but in the way that it works, it will still come into the observation and the rewards. So it should fit the framework, I think.
Okay. Okay. Well, it's something that I would need to look at more, you know, in more depth. But well, that's great. It's great. So it's a paper, the last paper I put up, right? That's the reference you mentioned that you were involved in. Okay.
Yes.
Okay. Fantastic.
Just a note on that. It was on an old version of Gama, so maybe we need some kind of update about it, but not that much actually.
Okay. Okay. Well, that's great to know. So that's very.
And just to add things, so there's a echo. Sorry. So from my point of view, you have two ways of doing, depending on the complexity of the reinforcement learning algorithm that you want to use. You can do directly in Gama, like I did for the very basic Q-learning algorithm. But if you want to use more complex algorithm, it can be better to use, like in the second work that you cited, to use external library for that we use here in Python, for instance.
I see.
So depending on what you do, I think we have several methods to do it.
Okay.
And for the second methods, we want to work on it in the future. So with Baptiste, so Baptiste makes the link between the Pythons and in Gama. And we are working with an expert in reinforcement learning, in particular in deep reinforcement learning, to be able to use it correctly in Gama. But I think in the future, we will try to have something more generic with the latest algorithm and so on.
Oh, that's not, well, no, I realized that you are the person who posted the, now everything makes sense. Everyone is here. That's so fantastic. Maybe I can reach, I don't know what's the best way. I can explore things and maybe at some moment have a conversation how all these new methods work. But yes, what you're saying is that because these things are very time consuming to execute, it would be better not to run them necessarily in Gama itself, but through some bridge, Python bridge, running them outside of Gama. Is that right?
The idea is maybe a bit about performance, but more about the tools, because as you know, the machine learning community in general is very
Python.
turned toward Python. All the tools are available, so we provided some kind of bridge in between.
I see. Understand. Yeah. Okay. It's more like to use the libraries that are available in Python because you
but
have
you
all
can
the machine learning
during the simulation. Yeah.
Okay. All right. No, this is great. And those things are not included or are included in the latest version of Gama. In particular, these type
no,
of
because
bridges.
it requires a little bit of how to say specific you have to describe some kind of protocol, what you want to exchange, what kind of data you want to exchange. So you can follow step by step, but you still have to do it a little bit by hand.
I see. Okay. Well, so work to be done then. Okay. Well, I hope this. I don't know if anyone has any other questions or comments. But this is great. It's great.
Other questions? It's been more than 20 minutes already, so if nobody has more questions, maybe we can stop here. In any way, thank you for taking time to come, even though you were like in between planes, and thanks to everybody for coming and presenting. As I said multiple times during the session, it was very diverse, and so kind of it was interesting basically. And maybe if Arnaud or Alexis wants to say a final word before we close the session.
So thank you for this session. It was a kind of a bit exploratory session. This session 6 was very interesting to see such a wide potential use of Gama. So thank you for everything for this session. Thanks again for everything. And Alexis, if you want to say a final word. Thanks also to all of course, the program committee, Baptiste, Patrick, Benoît, Arthur, Leo, Luca, Echi also for the co-organization, and Nicolas that I see here also. Alexis, if you want to conclude this, I think that can be nice.
Yes, thanks a lot, Arnaud. So it was a pleasure again being there, and very real pleasure this time because I didn't have to organize anything. So I would like to thank a lot, actually, Arnaud and Chi for the overall organization and making sure that the sessions were balanced and calibrated. So really there were really interesting debates and actually I think that's what makes these community so rich and probably what motivates us to develop Gama.
is diversity of applications and basically opinions you all have on how we can mobilize computer tools. Okay, so not I would say tools that normally we consider as very simple tools, how we can mobilize them to understand and maybe deal with the complexity of the world in which we live together. So for us, Gama has been, and it's still, of course, an instrument to talk about how we can tame complexity in trying to reach sustainable solutions to development problems. So mainly we are dealing with this kind of issues in the core set of developers of Gama. But actually, when I look at everything that has been done so far with the platform, it's really amazing, and it's always a pleasure actually to see this. For example, it's the first time I see a ping pong game made in Gama, and it has been something quite incredible to look at. Okay, although of course it's not completely revolutionary application, the way it has been done opens incredible perspective on how to mix the physical and the software world. And this brings me to the second thing, which is that actually we are all gathered more or less by the fact that we are using or want to be using a platform. But actually what's also very quite incredible here is the diversity of the tools everyone uses. So Gama more or less for most of you actually, when I look at what you produce, Gama is either the place where you are able to gather a number of tools and a number of practices, or something that belongs to a tool chain of your own. And this is for me also quite important, because we are probably not anymore in the platform does it all way of producing tools. Okay. And it's good, and actually it's very rich to see Gama being used with Python, with JavaScript, with being used as a server, etc., etc. Okay, so it's really a demonstration by basically that the tool in which we all invest, so this platform in which we all invest, will probably continue to be used for quite a long time, which is a good thing. And I hope next year, so I don't know, we didn't really talk about the next edition of the Gama Days, so maybe next year or in 2 years, I don't know. But I hope that next time we'll see even richer and more interesting applications. So thanks a lot to everyone. Thanks a lot, especially to the organizers, and of course to all the people who took their time to present their work and to answer the questions. Thanks a lot.